Hello, I'm Noel Hutchison. I have been working with Dr. Drew Pardall's lab to study the role of neurotrophic factor Nrn1 in AMPA receptor signaling and T cell tolerance. So T cell tolerance is the mechanism by which the immune system regulates itself to deactivate or delete self-reactive T cells that have the ability to target and destroy self tissues, resulting in autoimmunity. This is also an important process to understand because some cancer cells have the ability to hijack these mechanisms, resulting in the deletion or deactivation of T cells that are responsible for the anti-tumor response. Anergic or inactivated T cells have also been found in the tumor microenvironment, which is one of the main barriers to immunotherapy, CAR T cell therapy. So the functions of Nrn1 and AMPA have been thoroughly and separately investigated in the nervous system, though they have now been found to play roles in T cell tolerance. In the nervous system, Nrn1 is responsible for neurogenesis, the recruitment of AMPA receptors to synapses, and increased phosphorylation of AKT and mTOR, which are important markers for metabolism and nutrient uptake. In the immune system, Nrn1 has been found at increased expression in anergic and Treg cells and has also been seen to promote T regulatory function. In a Nrn1 knockout, there is worsened experimental autoimmune encephalomyelitis, which is the mouse model for multiple sclerosis. There's also decreased proliferation of Tregs, which may explain the worsened autoimmunity, and then also decreased phosphorylation of mTOR and changes in membrane potential. AMPA receptor is an ionotropic glutamate receptor with 4 subunits, GRIA1, GRIA2, GRIA3, and GRIA4. It has many accessory proteins, including Nrn1, and is highly expressed on postsynaptic neurons. The signaling of glutamate, a metabolic product, through the AMPA receptor allows the ion flux of sodium, potassium, and calcium. So we have to understand how Nrn1 interacts with AMPA receptor to promote Treg function. And based on the findings reported previously, we predict that Nrn1 acts through AMPA receptor to alter membrane potential and nutrient uptake via ion channel regulation. So in order to approach this, we first cultured CD4 T cells and differentiated them using cytokines and exposed them to either media with no glutamate or different concentrations of glutamate. The supernatant was collected from these cultures where glutamate was then measured. The cells were also sent through flow cytometric analysis. Then used co-expression analysis to assess if there were genes that were co-expressed by both Nrn1 and AMPA receptor. And then gene ontology analysis was used to assess the general function of those genes. And then in order to assess the function of Nrn1 in T regulatory cells, we used bulk RNA-seq analysis and R. So first we found that glutamate is secreted by T helper cells at different concentrations depending on subset. Not only does this mean that there is glutamate available for AMPA receptor signaling, but there's also glutamate available at different concentrations depending on the subset. Also found that the addition of glutamate to culture does affect the differential CD4 positive T cell proliferation and function. So different amounts of glutamate have different effects on the various CD4 positive T cell subsets. We then found that there was an overlap in genes co-expressed by both Nrn1 and AMPA receptor subunits GRIA2 and GRIA3. And upon gene ontology analysis of these genes in the overlap, we found a shared molecular function in ion channel activity. So in order to understand how Nrn1 and AMPA receptor work together to support T cell tolerance, we first have to understand how they work separately. So in a Nrn1 knockout, we found the decreased expression in genes specific to MHC and transmembrane complexes in Tregs. So using a gene set enrichment analysis in R, we looked at the biological processes that were activated and suppressed in the Nrn1 knockout versus the wild type, and found that Nrn1 is necessary for activation, proliferation, signaling, and transport in induced Tregs. Here is a gene set enrichment map that shows the groupings of these biological processes, which are mainly immune activation, phosphorylation, and secretion. So secretion may indicate an involvement in nutrient transport, and phosphorylation may indicate an involvement in signaling. Then the molecular function revealed that Nrn1 is necessary for cytokine signaling and receptor activity, immunospecific binding, and ion channel activity in Tregs. So here again is the enrichment map, but also we can see ion channel activity is highlighted as well. For the cellular component, Nrn1 is necessary for complexes within the plasma membrane of induced Tregs. So this is especially relevant if we are talking about changes in ion channels and membrane potential that are dictated by transmembrane proteins. Again, highlighting that there are both internal and external plasma membrane cell components that may rely on Nrn1. So in conclusion, we found that glutamate is indeed secreted and therefore available for AMPA receptor signaling, that glutamate does affect function and proliferation of T cells, that Nrn1 and AMPA receptor co-expressed genes are involved in ion channel activity. RNA-seq confirms that Nrn1 does drive Treg activation proliferation, but also Nrn1 knockout suppresses genes involved in ion channel activity. So ongoing, we're going to look into Nrn1 overexpressed in Tregs and Th0s, as well as an AMPA receptor knockout in Tregs and Th0s. And then in the future, we'll do a double knockout of Nrn1 and AMPA receptor. There are also calls for a further investigation into the specific effects of Nrn1 on ion channel activity. Thank you.